Okay, so just kind of off that idea, right? We have prophase, metaphase, anaphase, telophase. Those are the stages of mitosis. You should be familiar with those, right? And I'll kind of run through it briefly again. But you should know just the key events of each one. I'll leave these up here, so if you want to jot these down later, I just kind of scribbled them down real quick. But they're very easy to search up, very content-like. Nothing too crazy here. The real focus of the cell division chapter is not about the actual stages of mitosis. We should know those, but it's more focused on the regulating of the cell cycle. So the first question is, why do cells divide? Why are we made of trillions of cells instead of just thousands of larger cells? We've talked about this before. You did a lab on this. Yes?
Surface area of the volume.
Very good, because of surface area to volume ratio. The larger the cell gets, what happens to surface area to volume ratio? It increases or decreases? It decreases, which means it's harder for the cell to move the nutrients in and the waste materials out. The concept I kind of was thinking about is this, right? If you have your garden hose, the ratio of your garden hose to a little kiddie pool is a fine ratio. No problem. That's like a small cell. You can fill the kiddie pool. If you're trying to fill an Olympic-sized swimming pool with the garden hose, right, it's going to take you an extremely long time. You can't move enough water. The garden hose relates to the size and the ratio of the cell membrane to the volume of the cell. Right, so the kind of ratios there. What's the other reason for cellular division? It's not just surface area to volume ratio. There was something else I mentioned. We didn't do a lab on this part, but I mentioned it. Not really. The larger the cell, the more organelles. The larger the cell, the more chemical reactions. The more chemical reactions, the more what? We need to control them.
Oh, energy.
Energy, but that's more taking materials in and out. What controls chemical reaction? What? Enzymes. What has the information to make proteins, to make enzymes? DNA. So remember, DNA overload was the other one. DNA overload. So surface area to volume ratio and DNA overload. So DNA overload is simply saying that there's too many proteins that need to be made, and there's not enough DNA to keep up with that demand. You have a set amount of DNA, you can't access all of it at the same time. Right? It'd be like our Fort Lee library trying to service all the people that live in Manhattan. Can't do it. There's just not enough DNA there, not enough books there to carry that out. Okay. So that's just kind of our starting point. So why do cells divide? And now we're going to go into some of the details of it. Bring that back up. Let's see if our technology is actually going to work today and we can present something. Did I mention the Google Cardboard in this class? No. I'll mention another one. We might get a set.
Yeah.
VR viewers.
Actually, I have two of them. Well,
How do you get one?
yeah, it was
You
actually.
can buy them online. Don't buy the cardboard one though. I went online. I just bought the cardboard. It was like $20, and I realized if I looked a little bit deeper, there's nicer like plastic ones. You can get. I just bought one for my brother-in-law for Christmas. It's a VR viewer. It was $39, and it included a Bluetooth remote. So therefore, your cell phone slips into the viewer, and it's virtual reality. Looking around, like I got my Google Cardboard one yesterday. So my son Riley was in our bedroom looking around. He was actually like looking around the Museum of Natural History, like looking at the T-Rex and then turning around and looking at the Brontosaurus and Stegosaurus and walking around and seeing everything.
Yeah,
It's pretty cool.
Already, well, first of all, in terms of Biz will bring this up quickly.
Yeah.
A lot of companies are actually jumping on the virtual reality bandwagon. I've already seen. I've already voluntarily sat through two advertisements which used virtual reality.
Yeah.
The first was like a tour or whatnot of a. It was like a first-person tour of a
That's cool.
car.
Yeah.
The other one was for Clash of Clans.
There you go. It's pretty cool. It's amazing. I'll talk more about it another day. But like Xbox, PlayStation, as you know them, they're gone. That technology has passed. Right? Not that PlayStation can't take over virtual reality, but that's how simple and amazing this was with my son yesterday with the VR system in your cell phone. It's pretty cool. Any case, so we need to talk about cell division today, getting back to what we're focused on. Here are some key terms for cellular division. We have asexual versus sexual reproduction. The definitions people fall back to is actually a wrong definition, is the idea that asexual reproduction is one parent, sexual reproduction is two parents. That's not the way to think about it. Asexual reproduction involves more of a mitotic process, simply cells dividing. Sexual reproduction involves meiosis, the formation of gametes. Right? The reason why I don't think about one parent is because you can have a plant self-pollinate, but if the plant is forming gametes undergoing meiosis, it is considered sexual reproduction. So that's just kind of a concept we're introducing. Chromosomes, we know what chromosomes are. Chromatin, we've kind of talked about this already. We'll keep this down, and I'll write on the sides. So chromatin is the less condensed form of our DNA. It's still highly condensed. Remember, you have 6 feet of it in each one of your cells. But the chromatin form is kind of like an unwound ball of yarn all bound together. That's the form that your DNA is typically in. However, when it's time to assort DNA for cellular division, right, we don't want to keep it in chromatin form. We want to condense it. Nice, neat packets, easy to move around. So the chromatin condenses into chromosome form. Everyone okay? Blast in the past, regular vial, good. Okay, next thing we need to deal with are homologous chromosomes and sister chromatids, right? How many chromosomes do we have? 46 or 23? 23 pairs, right? 23 came from your mom, 23 came from your dad, right? There's the paternal chromosome. There's your maternal chromosome. Now, when your DNA is just hanging out, are the paternal and maternal chromosomes hanging out in chromosome form? No, they're in chromatin form. They're all over the place. But to show this, it makes it a lot easier to put them into chromosome form, just so we can see it condensed. Are these two chromosomes identical to one another? No, they're not identical, but they are what? Homologous, right? Which means similar in size, shape, information, but not identical. All that means is this. If I'm looking at the chromosome for eye color, the chromosome for eye color is going to be should be in the exact same position on both the maternal and the paternal chromosomes. Okay? So there's the gene for eye color. However, you could have two different, what's the word? Starts with an A. Alleles, right? You can have two different alleles where the allele paternal might be blue and the allele for maternal, the allele for maternal might be green. Okay, so that's what we mean by homologous. They're not identical, but they are similar to one another. Okay, now, before your cell undergoes mitosis, before your cell divides, what does it need to do with its DNA? Does it during S phase? It replicates our DNA, right? It replicates the DNA. When we replicate our DNA, this is what happens. Each one of these chromosomes forms a copy. Oh, sorry, I want to do that. Let's say blank. Okay, so here are my questions. How many chromosomes are present at the top? 2 chromosomes, right? Because that's a pair of homologous chromosomes, right? That's one of the 23 pairs, so there's 46 chromosomes, so there's 2 chromosomes there. How many chromosomes are present here? There's only 2 chromosomes present here as well. Now, we obviously have doubled our amount of DNA, so what's going on here? I'm not telling you, I'm not playing a magic trick, right? The DNA is doubled. So what's happened is we've doubled our DNA, but what we have here are 2 sister chromatids. Right? The reason why they're not considered 2 separate chromosomes is because those 2 chromatids are what to one another? I just copied them, they should be identical, right? So here I have two sister chromatids, right? Because they are identical to one another. So because they are identical, we still just consider them one chromosome. Okay. So here I have two chromosomes, but I have two sister chromatids here, two sister chromatids here. So if I look at this, how many chromatids do I have down here? The answer would be 4. I have 4 chromatids, right? Because I've doubled my DNA, but because they're identical, I still consider them just one chromosome. All right. So we will deal a lot more with this when we get into Mendel, probably sometime in the middle to the end of next week. But it would just need a little bit of basics to move through here. Somatic cells. What are somatic cells? So I'm giving lots of definitions and defining terms, and probably be a good thing to jot down some of these definitions, defining terms, unless you're going to spend
like any cell
20 minutes tonight, defining the terms on your own. Okay. So somatic cells. And what are somatic cells? Yes.
They're not like sexual or like sperm and egg.
Very good. Somatic cells are all the cells in your body that are not sex cells. Right, so that are not cells that are either gametes or going to become gametes. That's it. So all the cells, all your cells are somatic cells. Your skin cells, your neurons, everything's somatic cells. Now, when we talk about cells, we compare somatic. We also compare So we talk about somatic cells. We could describe them in one, we have 2 terms to describe cells, diploid and haploid. Which term would relate to somatic cells?
Diploid.
Diploid relates to somatic cells. Right? What do we use to represent diploid cells? What notation? Diploid cells are considered 2N cells, right? Because they have a full set of DNA. Haploid cells, gametes, are considered haploid, which are considered just N, just half a set of genetic information. All right, so we have diploid and we have haploid cells. Now, we use the word diploid, haploid all the time. The diploid number for human cells, right? We have 46 chromosomes. The haploid number for human cells is 23, right? But why do we use this 2N and N terminology? Because it makes it easier to deal with the diploid haploid numbers. Right? Think about having to memorize what the diploid haploid numbers for all these different organisms. Right? Carrots have 16 chromosomes or something like that. King crabs, how many chromosomes do you think king crabs have? They have about 216 chromosomes, right? So all different organisms have all different diploid haploid numbers. So it makes it easier to deal with a system that just allows us to deal with diploid haploid. Hence, we have 2N and N. What does N stand for in humans? What's that? 23. So N in humans stands for 23. When we form our sister chromatids, we have a nice little protein that holds them together. That protein is known as a centromere. Very good. The process we're talking about, which is division of the nucleus, is known as mitosis. So what I just said is the centromere is holding the 2 chromatids together. The kinetochore is involved with the spindle fibers, which I'll get to. So hold on, you're right. So one thing to understand, mitosis is division of the nucleus. The cell does not need to divide. You can have a multinucleated cell and mitosis can be complete. However, during telophase of mitosis, typically you have cytokinesis beginning, which is actual division of the cytoplasm, division of the cell. Now, the term that we might not have mentioned or heard of before is the kinetochore. The kinetochore is the region that is outside our sister chromatids. So here's our pair of sister chromatids. There's our centromere. The spindle fibers that come in we usually told that they just connect to the centromere. They actually don't. They connect to the kinetochore. And the reason why it's a little bit different is we typically envision the chromatids when they're pulled to opposite sides. We envision kind of the centrioles or centrosomes pulling on them like somebody fishing in or reeling in a fish. That's not typically how it works. It's more of the chromatid itself pulling on the rope and walking itself down the spindle fiber. That's what the kinetochore does. The kinetochore is where the spindle fiber attaches, and it's what's actually pulling the chromatid towards the pole. So it's more of the chromatid pulling itself, not the chromatid being reeled in, is the action. So spindle fibers, right? What are spindle fibers made of? Microtubules, right? The area in which our spindle fibers are formed is known as centrosomes. Centrosomes are present in both plants and animal cells. So centrosomes are the region where these mitotic spindles are formed. However, we also associate kind of the microtubule structure of centrioles, right, that are located in the middle of the centrosomes. However, those centrioles are unique to just animals. They're kind of an anchoring point for your spindle fibers, but they're not necessary, obviously, because plants do not have them. Do bacteria undergo mitosis? The question was, do bacteria undergo mitosis?
No.
Why not? They don't have a nucleus, so therefore, by definition, they can't undergo mitosis because mitosis is division of the nucleus. But they undergo a process that's pretty much identical to mitosis, right? Very, very similar. And that process is known as binary fission. So it's how prokaryotes divide. Everyone okay with some vocab terms? All right. Okay. So now that we're kind of familiar with vocab terms, we're talking about mitosis, we need to really dive into like the cell cycle, right? The life cycle of a cell. Interphase is the region or the part of the cell's life cycle when it is not dividing, it's not undergoing mitosis. Interphase makes up about 90% of the cell cycle. If you were doing your lab yesterday and you were including interphase, right, you would find that more than at least 90% of your cells were in interphase compared to the other phases you look for prophase, metaphase, and cells. So you weren't technically looking for interphase. One thing I don't like here is this schematic really implies that G1 phase, S phase, and G2 phase are actually similar in length. They're not. This pie slice, the G1 pie slice, should be much larger, and the S phase and the G2 phase should be much smaller. So just keep in mind that their time frames are not that close as this chart is kind of comparing. G1 is far longer than S phase or G2 phase. Oh, I don't want to erase that. Okay, so we start with G1 phase. Is the first phase of the cell cycle, right? Our cell has just divided. I have a brand new baby cell, tiny, cute little cell, right? What does that cell need to do? Well, it moves into G1 phase. So G1 phase is really growth and working. All right, the first thing that has to happen is that new cell needs to grow. Might need to make some more mitochondria, depending on what the function of the cell, might create some more membrane proteins, rough ER, smooth ER, whatever organelles that cell needs. It's going to grow and become a more mature cell. But then this is the main working phase. So this is the phase where your neuron is being a neuron, right? Your skin cell is acting as a skin cell. Your red blood, well, let's not use red blood cells. Your white blood cells are acting as white blood cells, and so forth. So that's why this is by far the longest stage of the cell cycle, where cells mostly are hanging out because it's where they're carrying out their job. The next phase, S phase, is very simple. It's simply just S phase is simply just DNA replication. right? We are simply just replicating our DNA in S phase. At this point, if the cell moves from G1 phase into S phase, it is for the most part decided to divide. It's decided to divide. That brings us to our last phase, which is G2 phase. G2 phase we kind of label as the checkup phase. Right? We've now replicated our DNA. We're getting ready to move into mitosis. However, we just need to make sure that everything is correct. Like the DNA is replicated properly. There's no major mutations. Why we want to make sure everything's there. Our centrioles are formed. If we need to make any accessory proteins to go with mitosis. It is the checkup phase. So we're just kind of making sure we are ready to move into mitosis. If we're good with G2 phase, the cell moves into M phase. M phase includes mitosis and cytokinesis. Just realize that M phase is not mitosis. M phase includes mitosis and cytokinesis. Cytokinesis is not part of mitosis because that's a division of the cell. Mitosis, again, is just division of the nucleus. Everyone okay? Kind of redundant. You've had this in bio class before? Yes? No? Yeah. Okay, good. Okay. Then we go into mitosis. I'm not going to go through this again. We've kind of walked through and talked about the key things. So just to be familiar, you know, like the main key events of mitosis. The one thing I just want to mention to make sure you guys are familiar with it is the cytokinesis process, right? During telophase, your cells are going to divide, right? They're going to split. They split in two different ways. If you have an animal cell with no cell wall, right, you have a cleavage furrow that forms. So your cytoskeleton can help shape the membrane. It starts pulling in on the plasma membrane and causes a cleavage to form. It pulls those two plasma membranes together, and eventually that one cell will divide into two, being controlled by your microfilaments and your microtubules. Plant cells can't do that because they have a cell wall. So plant cells do something a little bit different. They form what's known as a cell plate. Right, they form what's known as a cell plate, which is formed of all these lipid carbohydrate molecules. So these are carbohydrates surrounded by a lipid membrane. Those start to fuse together, form a cell plate. The carbohydrates start aligning and connecting down the middle with the lipid bilayers forming the outside. When you're done, you end up with a cell wall and a newly formed membrane. So it just forms cytokinesis plates, just formed by cell plates. Okay. Binary fission, just kind of relating it to this process to prokaryotes. Right? They don't undergo mitosis. There is no nucleus. Couple key things here is one, how many chromosomes do we have? 46. Right? We have a number of different chromosomes. Prokaryotes, how many do they have? They simply have one. They have one unique chromosome. So what happens is this process is you have your prokaryote. Here's my chromosome. DNA replication starts at one point, and it just starts replicating its loop. Once you're done, you end up with simply two chromosomes. All right, we just need to finish up the replication right there because we're just replicating around the loop. We replicate our second chromosome. Once that happens, they just move to opposite sides of the cell and the prokaryote divides. So it's not really any different than mitosis. It's just kind of terminology because there is no nucleus. This is just kind of looking at binary fission. All right, so we have our two copies. Right, our copies being made, right? Our copies are simply shifted to opposite sides of the cell, and then that cell divides. Okay, this is what we really care about. This is what we're trying to rush to get to, right? Trying to get through the content that was kind of basic stuff to the actual regulation stuff. We're going to talk about internal regulators. We won't necessarily get to the external stuff today. We're going to kind of finish with the internal regulators. So what we have here is we have two different molecules, right? We have cyclins and we have CDKs, cyclin-dependent kinases. We've mentioned kinases before. Right? What is a kinase? What do kinases do? In fact, you probably have it because it's actually in the case study you're doing, so you might not have gotten to it yet. But as you read about the case study, kinases are simply enzymes, and they're enzymes that phosphorylate things. Right? So they phosphorylate proteins. And typically, when you're phosphorylating something, you are activating. However, sometimes something could be phosphorylated to inactivate it, but it's more likely to be activated. Right? So kinases are enzymes that activate other proteins, activate other enzymes. What we have is a group of kinases known as cyclin-dependent kinases. These cyclin-dependent kinases is what regulates your cell cycle. Okay. Proteins, they're large, complex molecules. Do we want to build, construct, synthesize, and destroy proteins all the time, or do we want to keep them somewhat intact? Yeah, we want to keep them intact, right? Because there's a lot of energy, there's a lot of work that goes into building these proteins. So how this system works is your CDKs, your cyclin-dependent kinases, they are formed, and for the most part, they stay consistent. Their numbers stay consistent in the cell at any part of the cell cycle. So what we need is specific activator molecules to turn them on, turn them off, right? We don't want to build these big, expensive proteins. We want to build these simple, cheap, effective activator molecules. Those activator molecules are cyclins. So cyclins are small, little activator molecules that bind to the CDK enzymes to simply turn them on. So the cyclin CDK concept is simply just activators and enzymes. Okay, so we will get back to cyclins and CDKs in a second. But let's talk a little bit more about the cell cycle. We said, again, the G1 phase is much larger, so don't let the size of the pies confuse you. But cells spend most of their time in G1 phase. And what they have is they have what we call a checkpoint. We call it the G1 checkpoint. And what that G1 checkpoint is doing is it's ensuring that cells are locked into the G1 phase. Right? The G1 checkpoint will not allow the cell to progress past it unless certain environmental factors are present. So that is our G1 checkpoint. When those environmental factors are present, the cell will be allowed to move from G1 into S phase, right? Then we start replicating our DNA. We also have our G2 checkpoint. The G2 checkpoint is a checkpoint to make sure that everything is all set. All environmental factors need to be okay, need to be present to allow the cell to move from G2 phase into M phase, into mitosis, to divide. Then we have the M checkpoint. The M checkpoint is simply the checkpoint allowing it to move from mitosis right into cytokinesis and back into the G1 phase. So these are the major checkpoints of the cell cycle. What regulates them are the cyclins and the cyclin-dependent kinases. So just kind of a quick example on the left-hand side here. This is just a little experiment they did. So here I have a cell that's right in the middle of mitosis, right? So there are going to be specific chemical characters that are present in that cytoplasm right now. Certain cyclins are going to be present. If you extract the cytoplasm out of there and inject that cytoplasm into a cell that's just hanging out in G2 phase, because those cyclins are present, the cyclins for mitosis are present, that new cell will automatically jump into mitosis because it's simply reading chemical factors. Chemical factors are there, it's going to follow along and jump through that checkpoint. Okay. So, this is what's going on. You do not need to know cyclin 3, cyclin 4, cyclin D, cyclin A, cyclin E, CDK1, CDK2, 3, 4. You don't need to know any of those numbers. You don't need to know any of the specific cyclins. All we need to know is kind of how this process works. Our CDKs are present all the time. And simply, when your cell needs to progress through particular checkpoints, your cell starts producing the cyclins, the internal regulator factors that allow it to move through that checkpoint. So here I kind of have our G1 checkpoint, right? In order to get through the G1 checkpoint, CDK4, CDK6 need to be activated. Well, when environmental factors are ready, right, your cell will start to produce cyclin D. Cyclin D is a very small, easily produced molecule. Cyclin D will bind to the CDKs, activating those CDKs, allowing that cell to progress through the G1 checkpoint. Once you're through the G1 checkpoint, it's easy to degrade, to destroy these cyclins. You can start destroying cyclin D, and as you start replicating your DNA, your cell will also start expressing or forming these cyclins A, cyclin E's, right? Allowing to activate those specific CDKs to allow us to move from S phase into G2 phase, and then so on, right? You're going from G2 phase into M phase, right? You might have the continuation of cyclin A, but then the formation of cyclin B, right? And the activation of those specific CDKs. So how you need to be able to handle this is simply, right? The rising and falling levels of cyclins controls the cell's process through the cell cycle. Specific cyclins need to be present in order to move through the G1 checkpoint. Those cyclins can then be destroyed, and new cyclins can be formed to allow the cells to move through the S phase, the G2 checkpoint, or the M phase checkpoint. Right, so there's not specific names you need, just kind of more of the rising and falling levels of cyclins. This relates directly back to what we just talked about, which is signal transduction. Remember, signal transduction relays outside messages to cause cellular responses. Right? Hormones are oftentimes involved. Well, hormones are oftentimes the ligand in this process. So I have HGH hormone binds to the tyrosine kinase receptor. Okay? The message is transduced, relayed through the cytoplasm to the nucleus of the cell. Right? And that message causes a cellular response, and that cellular response might be, hey, we need to progress through the cell cycle. We need to move from G1 phase into S phase. So specifically, that cellular response would be telling the nucleus to start making what? Cyclin D, right? Relating the signal transduction to an actual cellular response. So how the external factors can control the internal factors, and so forth. This is just another look. It's really the same thing. It's rising, falling levels of cyclins. I just put it up there because it's here, I guess, but I guess the other one's better. It's in French, so that's fun. And the one last thing is this. You don't need to know specific cyclins, but there's actually one that we do need to be familiar with, and it's known as the MPF. It's the maturation promoting factor, the M phase promoting factor. Right? That is the specific CDK cyclin complex that allows the cell to progress from G2 phase into M phase. So the MPF is simply a cyclin CDK complex, and it's what is present that allows the cell to move through the G2 checkpoint. It's either called the maturation promoting factor or mitosis promoting factor, MPF01. So that's the one you just might see, and that's just the cyclin CDK complex that allows it to progress through G2. Now, why is this so interesting? It's because if we truly have a grasp and understand how to control this process, what can we wipe out? Mutations are part of it. If we learn how to really, truly control this process, right, in every type of cell, we can totally wipe out what disease? Cancer, right? Cells that are cancerous have lost the ability to control this process. They are moving through the cell cycle without regulation. So really understanding what's going on in the individual cell and how to actually control this process in individual cells is really one of the methods to kind of solving things. We will talk more about cancer tomorrow, but just kind of realizing that that's the bigger implication of understanding this process.